what does this mean for natural populations? And I struggled with that myself in trying to decide how I could use these results to have general evolutionary consequences instead of specific only to my population. So what I've done is focused on the genotypic values that Charles Goodnight talked about, the additive genotypic value, the dominance genotypic value, and as we'll see today, epistatic genotypic value. These values are merely the differences in means between different genotypes. So they don't involve gene frequency at all. The gene frequency in my population happens to be 50%, which is very convenient because it gives you the most power to detect genetic effects. But we can take the results and project them into populations of any allele frequencies we wish. And you'll see examples of that today, where I take the results from our study population and then project them into situations that are probably more realistic in terms of natural populations than all alleles being at a 50% frequency. So today we'll talk about epistasis. As Dr. Goodnight gave an excellent introduction to the idea, and you'll see some of these ideas repeated. You'll see some of the virtually the same slides. I come at it from a slightly different direction, and you know but everything is actually the same. I'm not going to contradict anything that he presented. So if it seems like I am, please ask me. So last time we talked about many genes of small effect with a variable role of dominance in the genetic architecture. So today we move on to do gene effects vary depending on the genetic background. The genetic background are all the other genes present in the individuals. And that variation depending on genetic background is referred to as epistasis. So I want to stress that the effects of genes are not properties of the gene itself, but are properties of the gene in its context. So you say a gene causes multiple sclerosis, and you'll see that typically that gene does cause multiple sclerosis, or that allele does cause multiple sclerosis, but in some individuals carrying the allele it doesn't. And there can be a wide range of variation in the severity of multiple sclerosis, even from the same allele. And this is due to contextual effects, the context changing the effect of the gene. So even a severe disease seen as monogenic can actually have a polygenic basis and be affected by epistasis. So the effect of a gene is not an autonomous property of the gene, but depends on its context. And the context we talked about yesterday, we talked about sex, where male and female, genetics of male and female for body weight, obesity, and diabetes were very different for the males and the females. That it was hard to say what a gene would do unless you specified the sex. Likewise, we had multiple environments with the high and the low fat diet, and again, it was difficult to say what the effect of a gene was unless you also specified the diet. So today we'll talk about epistasis and gene interaction as another contextual factor. And we'll also then later talk about pleiotropy and what happens when you consider these issues in the light of multiple traits. Now, this context dependency of genes has important consequences for understanding the genetic basis of complex diseases, preferred treatments, and evolutionary processes. And we'll talk some about evolutionary processes affected by epistasis in the latter part of each of the lectures today. So the idea that things are context dependent and is not something that's special to genetics or evolutionary genetics. It's true in general. So here I have a quotation taken from Polybius, who was a Greek author who wrote about the Second Punic War. He was well, he wasn't a captive, he was a guest of the Romans. In other words, he was taken out of Greece and lived in Rome and actually hung out with all the top Romans and was in a good position to write the history. And this is his view of how one should look at the history. And it has always seemed to me that those who believe they can obtain a just and well-proportioned view of history as a whole by reading separate and specialized reports of events are behaving like a man when he has examined the dissected parts of a body once alive and beautiful, imagines that he has beheld the living animal in all its grace and movement. The fact is that we can obtain no more than an impression of the whole from a part, but certainly neither a thorough knowledge nor accurate understanding. We must conclude, then, that specialized studies contribute very little to our grasp of the whole and our conviction of its truth. So he's saying this in the context that he was writing a history that was full Mediterranean, the entire Mediterranean taken in together, rather than what happened in Greece, what happened in Italy, what happened in Africa. He was integrating all of these together because he felt that you couldn't understand the historical processes that were going on in Italy without understanding Greece and Africa and Asia. But the same principle holds. Just instead of history, put in genetics. So there are several reasons that you might want to study epistasis. It can help us understand heterogeneity in results from different populations with different gene effects. Under epistasis, you expect the effect of a gene to change depending on the frequency of other genes that are present. And different populations can have different gene frequencies at these other loci, thus modifying the effect of the locus you're interested in. It can help understand the potential physiological relationships amongst quantitative trait loci, QTLs. Those are the regions of the genome as we were looking at yesterday. In that if there's a statistical, or there's an epistatic interaction between the two genes, it implies that there is a physiological interaction, either directly with the two gene products interacting, or because they both operate in the same pathway and can affect the outcome of the pathway jointly. It can also help us to identify regions of the genome that only have a conditional effect on a trait, that have no main effect on the trait. When you do your scan through the genome, you don't see them, they're hidden, because their effects are conditional, and we'll see some examples of that. And it can also have important evolutionary effects related to population differentiation, hybridization, speciation, and also would say modularity and the structure of genetic architecture for complex traits. Now there have always been two ways to, well I guess there's two ways you can look at epistasis. Two ways you can try. One is to detect epistasis by measuring the interaction variance. Dr. Midnight talked about the interaction variance. This is the way people have usually looked for epistasis and haven't found much. There's a second method of looking at that, and that's to measure epistatic genotypic values that are free of the effects of allele frequency at the loci involved. You do this, you find, as you'll see, a whole lot of epistasis, and epistasis that has important evolutionary consequences. So when I say epistasis, I mean epistasis occurs when the phenotypic effects of a gene depend on the genotypes present at other loci. So you remember this from from Monday's lecture. This is our number line out of Falconer. We have the two homozygotes at either end. The midpoint of the homozygotes is set to 0. And the definition here of the additive genotypic value is half the difference between the two homozygote genotypic values. These genotypic values, you get everybody in the room who is a capital A, put them in a corner, measure how tall they are, and get the average of that. That would be the genotypic value for capital A. You get the small a's in another corner, and the heterozygotes in yet another place, and you just get their averages. So it's actually pretty simple. The dominance genotypic value is defined as the deviation of the heterozygote from that midpoint of the two homozygotes. And that's the second equation. Dominance can take on different values. We saw last time that about half of the loci we were looking at showed codominance, with the dominance value being 0 or not significantly different from 0. With codominance, the heterozygote is halfway between the two homozygotes. Or we could have capital A dominant to small a here. And in that situation, D is equal to the additive genotypic value plus A. Or small a could be dominant to big A. That's perfectly all right. I'm not using the tall capital and small letters to indicate dominance, and you'll see why after a bit, but it could be that the small a allele is dominant, and then D would equal minus A. You can't even have a situation where the heterozygote is less than minus A. So it's not between the two homozygotes, but smaller than either of them on this scale. And that's called underdominance. And likewise, you can have a dominance value greater than plus A. That would be overdominant, the heterozygote larger than either homozygote. So all of these possibilities exist, and actually in the examples yesterday we saw all of these possibilities. So for epistasis, we got to change the number line a little bit. And I'm sorry for changing symbols too. But, so if we look here at the top, we have two loci. One has two alleles, S and L, oddly enough, large and small, right? And the other locus has got X and Y as its two alleles. So the top number line is like what you just saw. We have the small homozygote, the heterozygote, and the large homozygote lined up. And the heterozygote is midway between the two homozygotes. So this, if you have the XX genotype at the second locus, then this is the relationship of the S and L alleles. It's codominant with the small allele making you smaller and the large allele making you larger. However, under epistasis, if we go to another genotype at that XY locus, we see that the relationships amongst the genotypes changes at the SL locus. So instead of a purely additive codominant situation above, here the two homozygotes are not different. They're the same, but the heterozygote is way out there. So this is a case of overdominance, and the large and small alleles have no additive effect if you're XY. Finally, you go down to the third number line, YY, and you'll see that this is just a mirror image of the first one, right? It's codominant again. The heterozygote is midway between the two homozygotes, but it's the small allele that makes you bigger and the large allele that makes you littler. So the A has switched, the additive genotypic value has switched sign between the XX and the YY. Whenever you have epistasis, it always has to involve both loci. Sometimes you'll hear people talk about a major gene and modifier effects. Well, if the major gene has these other loci that modify its effects, it also is modifying the effects of the other loci. You can't pull these loci apart and treat them separately. So anytime that there's changes in the relationship of the S and L alleles across XY genotypes, there's also going to be changes in the XY over the S and L genotypes. So here you can see, again, here X is SS, X is dominant to Y, and Y makes you bigger. SL, you got overdominance here again, and LL, you have Y dominant. Actually, these lines do correspond to the ones on the earlier slide. They're showing the same numerical values, just now we've drawn the lines for a different locus. It's important to know that it's not a one-way street. Epistasis always goes both ways. So like we had the genotypic values, the additive and dominance genotypic value at a single locus, we can define genotypic values at a pair of loci. So here we have locus A with a 1 and a 2 allele, and locus B with a 1 and a 2 allele. And then each one of these categories is a genotypic value for the individuals carrying that genotype. So we get everybody that is A1, A1, B2, B2, put them in a group, get the average, and that's the number that goes here. Again, very simple math involved. So now, when we define our additive genotypic value, we have to define it depending on the B genotype, because you know it's going to change. So here we see the first row, the additive genotypic value in the first row at the A locus, the additive genotypic value in the second row, and the additive genotypic value in the third row. And basically, you just average them to get, divided by 6, to get the overall additive genotypic value for locus A. To get the additive genotypic value for locus B, you would simply do the same thing with the columns. It would be this one minus that one, minus that, and finally the 2, 2, 2, 2, minus 1, 1, 2, 2. Likewise, the dominance genotypic value is going to be the average of the 3 dominance genotypic values, one time for each row.
The G222 on the first equation should be a plus, right?
There, yes, that should be a plus. The 2222 should be a plus. You know, as I was telling you the other day, you start doing this stuff and the pluses and minuses kind of randomly change in front of your eyes as you glaze over trying to do the proof. That's why it's always better if pencils are not well designed, they don't have near enough eraser on them. I would always run out of eraser well before I run out of blood. So those are the genotypic values. And what we're going to do is divide the genotypic values into 2 different parts, an epistatic part and a non-epistatic part. The non-epistatic part accounts for additivity and dominance. The epistatic part only contributes to epistasis. So here you can see the equation for the non-epistatic values. The thing I want you to notice about these equations is they never have gene frequencies in them. I haven't got any gene frequencies here anywhere. I'm still just dealing with the means of groups. So it's critical, of course, that you know every stage of that, right? Just remember I can separate the epistasis from the non-epistasis. So each genotype has a non-epistatic genotypic value and an epistatic genotypic value, which is just gotten by subtraction. the full genotypic value minus the non-epistatic one gives you the epistatic genotypic value. And in this epistatic genotypic values, we have some constraints on these numbers, that each row and each column has to add up to 0. The non-epistatic values take care of the rest, the main effects that go across a row or down the column. Here it's only the epistatic effects. So since the rows and columns have to sum to 0, while we have 9 numbers here, there's really only 4 different numbers. If I know the top 4 in the corner here, I would be able to tell what the bottom row and the last column contain, because it has to add up to 0. So while there are 9 of these things, there's really only 4 of them. And in quantitative genetics, you divide it up in a peculiar fashion we'll talk about. We don't just use those 4. We use all of them, but that results in 4 independent numbers. So we can take these genotypic values representing epistasis and non-epistatic values, and in order to use them in a population context, in an evolutionary context, we have to put in gene frequencies. And this is done through the average effect of a gene. And this is an average effect of a gene substitution or an allele substitution. As Dr. Goodnight said, you take out the one allele and you stick in the 2 allele. What's the change? And in the definitions from Faulkner, you've got the average effect of an allele substitution is going to be equal to the additive genotypic value plus the dominance genotypic value. Ooh, got carried away. The dominance genotypic value times P2 minus P1. These are the allele frequencies of the the A1 allele and the A2 allele at the locus. So P1 and P2 are for the A locus, Q1 and Q2 are for the B locus. So you can see that this part here is the ordinary quantitative genetic definition of the average effect of an allele substitution that everybody's familiar with. And what epistasis does is it adds on another term to it. So this E11 dot dot is equal to Q1 squared times the 1111. Maybe I can't use the red dot without going crazy. 2 Q1 QQ, Q1 Q2 times the heterozygote of this first row, and Q squared 2 times the 1122 genotype. And these other terms would be the same. They would contain, instead of 1111, this would be 1211, 1212, and 1222 for this term. What's important to notice is that they contain these epistatic genotypic values, and they contain the allele frequencies, both at the locus of interest, the P1 and P2 here, and at the interacting locus. So now we've introduced allele frequencies into the situation. And the additive genetic variance, or the heritable variance, is simply 2P1, P2, average effect of an allele substitution squared. If we had the same, we do have the same equations for the B locus, and what you would do is calculate, alpha B the same way, different numbers with the same pattern. And if we wanted to get the total additive genetic variance, we would add the A locus to the B locus. You just add them together. The fact there's epistasis makes some people think you can't add them together, but that's not true. They're simply additive. like the average effect of an allele substitution, we also have the same process occurring for the dominance deviations that Dr. Goodnight talked about. And here, if you look at the first term before the plus sign, these are the standard dominance deviations from that least squares regression that we had before, and that you'll find in Falconer. However, we see that there's an additional term here, including those epistatic genotypic values. And then to get the dominance variance, this is what you'd have from Falconer, 2p1, p2, dA, the whole thing squared. And we've added in then an epistatic term. The interaction deviations, sometimes called the epistatic deviations, but I try not to do that so people don't confuse them with the epistatic genotypic values, because they are different things, are calculated this way. And I've used the Greek letter epsilon for them instead of the standard letter E. So you take the epistatic genotypic value at that location minus the average over the ij row minus the average over the KL row plus the average over all 9 values. While all of the epistatic values had to add together to 0, once you put in gene frequencies, they don't add to 0 anymore. And the interaction variance is simply the variance of those 9 values. And since the population is a deviation from the population mean, so the population mean of these values is 0. So out of all that, that's just to show that there's lots of equations. If you can remember this slide, at this point, it would be the main one. So we have the physiological component. These are the genotypic values for additive, dominance, and epistasis. And over here are the variances, the additive, dominant, and interaction variances. People have traditionally had looked for epistasis using this interaction variance. And of course, if there is epistasis, there will be an interaction variance. But as you saw from the equations, epistasis also contributes to the dominance variance and the additive variance. These components of the effects would be left out and not looked for, where in many populations, the major effect of epistasis on variance is through the additive variance or through the dominance variance, not through the interaction variance itself. So while variance components are the standard in quantitative genetics, if we look at these genotypic values, we get a different picture of the importance of epistasis. We'll just show you some of these kinds of epistasis with charts like this. This is an example with no epistasis at all. Here we have the genotypic values, the 9 genotypic values for the 2 loci together. And you can see if we go down this first column that A is equal to +1. Right? Half the difference, 0 minus a minus 2 is divided by 2 is a +1. Here in this column, basically, it's +1 as well. And down this column, it's +1. So the additive genotypic value from column to column doesn't change. Likewise, if you look across the rows, the genotypic value in each row is the same. It's all 1. So here there's no epistasis. And if we were to plot the phenotype along the y-axis and the genotypes along the x-axis, what you would see is a series of parallel lines, lines that would never cross. They're parallel to one another. That's the situation of no epistasis, which is indeed a very special situation. So I said that there are really only 4 separate numbers for the epistatic genotypic values. And this is the way they're usually divided. And this division actually has, as we'll see, there are different evolutionary consequences for the different kinds of epistasis. So this is additive by additive epistasis. You can see in the first column, A is +1. The second column, A is 0. There's no differences. The third column, A is -1. So the additive genotypic value at the second locus over here changes from +1 to 0 to -1 as we go across the genotypes in the columns. And the same thing happens to the rows here. We have a +1, 0, and -1 additive genotypic value. When you plot these on a chart, you get phenotype here, genotypes along here, you get a star. Here, the 2 columns and rows have crossed. This represents the heterozygous genotype here, where there's no effect at all. So unlike the parallel lines, here we have crossed lines. That's epistasis. Now, as we talked about, epistasis can cause additive variance, dominance variance, and epistatic variance, or interaction variance. So here we have the additive by additive epistasis can produce epistatic variance. Can produce epistatic variance. That's shown here, the interaction variance. P and Q are the gene frequencies at the 2 loci. Remember, P was for the A locus and Q was for the B locus. So we've only got 2 alleles here, so the 2 alleles are P and 1 minus P in their frequency, or Q and 1 minus Q in their frequency. And you can see that epistatic variance is highest at 50%. When the allele frequencies are both at 50%. However, additive by additive epistasis does not produce any dominance variance at all. But instead it does produce additive variance. And you can see at the midpoint here, at the 50-50 point, the additive variance produced by additive epistasis is 0. And then it goes up on either side. So you get 4 different peaks, 1 on each side, and those peaks refer to the situation where the A allele is fixed and the B alleles are intermediate in value, or the B alleles are fixed and the A locus has an intermediate frequency. You also should notice that these are drawn to scale so that the highest additive variance you can get out of this epistatic system is double the maximum of the interaction variance. And that was all consistent with what Dr. Goodnight showed. We also have additive by dominance epistasis. So in this kind of epistasis, the additive genotypic values change at one locus across the other locus's genotypes, while the dominance changes at the second locus. We look at this now, you have our first column, we have a +1. The second column, we have a -2 as our additive value. And in the third column, we again have +1 again. So here, the additive is changing at this locus, while the dominance changes at the loci across the top here, where it would be overdominance. The heterozygote is +2, while the homozygotes are -1. So that would be a +3 overdominance. And then in this last row, you have underdominance. And again, here's those lines drawn, and you can see that there's crossing, they're not parallel lines. So this is another form of epistasis. And it can also produce epistatic dominance and additive variance. And this shows the plots with the different gene frequencies. You see the epistatic variance again is at its maximum when P and Q are both about 0.5. The dominance variance is quite high if you're fixed at the second locus and you're intermediate at the first locus, or vice versa, for either allele at the first locus. And it can produce amounts of additive variance as well. Again, these are all drawn to the same scale, so you can see the amount of additive variance it produces is potentially much less than additive by additive epistasis. It's an important distinction between them. This is additive by dominance epistasis. There's also dominance by additive epistasis, which is the exact same thing with the loci transposed, where the second locus has its dominance change while the first locus has its additivity change. Now this is dominance by dominance epistasis, and if you look down here, we have underdominance in the first column, overdominance in the second, underdominance in the third. First row, underdominance, second row, overdominance, third row, underdominance. And here again are the lines, and they're certainly not parallel. These two have been displaced from each other. You can see, so you can see there's two lines. They're really right on top of each other. And even this form of epistasis, as you can see, it can produce lots of interaction variance. It can produce fair amounts of dominance variance, both when they're 50-50 and when one locus is fixed and the second is variable. And likewise, it can produce additive variance as well. But again, a relatively small amount compared to additive by dominance, and an even smaller amount relative to additive by additive. So I'll talk now about QTL mapping. How do we find epistatic loci? So you remember these guys. We're going to do this population again. And we're going to focus first on the adiposity loci for the reproductive fat path that we talked about some yesterday. And this is going to be based on the F2 population. So to remind you, here we had effects differing by sex, 4 QTLs affecting males, 1 affecting females, 3 affecting both sexes. All of them have pleiotropic effects on overall body weight, but only 2 also affect skeletal size. And here what we're going to see is that the effects of these QTLs depend on genotypes and other loci. So this is a chart, a network of the epistatic interactions that's just between the QTLs that we saw. You can see that has all kinds of epistasis represented, additive by additive, additive by dominance, dominance by additive, and dominance by dominance. All of them are represented in here somewhere. And that adiposity 8 is central to the epistatic interactions. It interacts with every other locus. The other loci also interact with someone else, so additive 2 interacts with additive 3 and additive 8. 5 is one that just interacts with additive 8. 1 interacts with 3, 6, 7, and 8. So it's a pretty dense network here with a lot of connections. And this is just the genes that we saw because they had a non-epistatic effect as well. So this is an example here of the genotypic values in a situation where there's epistasis. D18 MIT51 is the name of a microsatellite that sits right at adiposity 8. So this is adiposity 8. This is another locus on chromosome 18 on the proximal end of the locus. And it shows a strong interaction with adiposity 8. You can see that there's an additive by additive interaction, and that if we look at this column of columns, it goes up as we move from the third genotype to the first genotype. However, in this last row here, 3 genotype makes you bigger. First row, smaller, second row, bigger. You can also see that if we look at it in the other direction, here we're stepping down as we go from 1 to 3. While here and in the yellow bars, we step up as we go 1 to 3. So this adiposity 8 showed up as a main effect. And if we look at the 3 different genotypes at the other locus, we can see why. It's going to be the effect that we see at adiposity 8 is going to be the weighted average of the effects in these 3 genotypes at the marker proximal to it. So if we look across this first row, we see there's some negative effect, but the effect isn't very big. So 25% of the population has basically a small negative effect as we go from 1 to 3 at the adiposity 8 locus. In the second row, though, you can see there's a really big effect here. The fat pad weight going up to 1 gram as opposed to 0.8 grams. And the third row, this is 50% of the population. And then the third row has, again, the same pattern, low, low, and then high. So actually the small allele, which is the 1 allele, is dominant in these rows, the yellow and the purple, but not in the blue. But 75% of the population shows an effect. So we see that effect when we ignore epistasis. The 25% of the small minus effect waters it down a little bit, but not very much. So just like we saw with the different diets and environments, diets and sexes yesterday, when there's interactions, you can wipe out main effects that you can't see because they're too small. Here, since 75% of the population has an effect at this locus and 25% doesn't, that's enough to see it. But your idea of how big the effect is that it'll be half of what it should be. If we look the other way, here, we can see that 75% of the population has little or no effect. And it's only if you're large homozygote at adiposity 8 that you see a big effect of this P18 MIT17 locus on obesity. This one you don't see in the genome scan. Because 75% of the population have no effect, only 25% does. And even though it's a really big effect that it has, it's going to wash out with the 75% null, and you're not going to see this locus. So epistasis can hide loci that have effects on traits. This is the beautiful equation which shows how you would go about calculating the additive by additive, additive by dominance, dominance by additive, and dominance by dominance values. Here's another definition for them, only using the means. You'll note that there are no allele frequencies in this definition. It's all with the means. One thing, like the minus sign was wrong before, this 9 is really an 8. Because I lost a degree of freedom making up the overall average of things. So that really should be 8. You can see that the t-values and the t-values for these epistatic terms contain the normal term you're used to for anything. You know, it's the value divided by the standard error, and that quantity squared and square rooted. Here we can see that the additive average effect of the allele shows up in the dominance by additives, and in the dominance by dominance, we have the dominance at both loci showing up, along with 1 minus the error correlation between A and the two loci, dominance values for the two loci. So it's a little more complicated here because this has to take account of the gene frequencies. Because when you're doing a statistical test, standard errors depend on how many individuals you have in the category. So for statistical testing, frequencies have to come in, but they don't affect the values themselves. So instead of doing a genome scan along one dimension like we did last time, here we're doing it along 2 dimensions. This again is chromosome 18 on the x-axis and chromosome 12 on the y-axis. The probability of an interaction is given color-coded here. And you can see that there seem to be 2 foci for epistatic effects. We have this is adiposity 8 over here, and it's interacting with something, partway down chromosome 12, about halfway. I guess it's a little more than halfway down chromosome 12. However, we also see another effect here. This is actually the location of that D18 MIT17. This is that second locus I was showing you. And here it shows up again, interacting with the proximal portion of chromosome 12. So you do this for all the pairs of chromosomes. You get a really big chart. So each of these would just look like a red dot with a yellow circle around it. And you can get it throughout the genome. When you go out throughout the genome, you find lots of epistasis. However, remember when you're doing a lot of tests, you're going to get significant results by chance. And if you're doing a lot of tests, you have to correct for that. And that's done a different way here. Only 1 or 2 of these beat the Bonferroni. But if we look at how many cases of epistasis we see versus how many we expect at the 5% level when there's really no epistasis at all, then we have an enrichment for epistasis. And as we limit the significance level further to 1% and 0.1%, the expected number goes down. Of course, as does the observed number. But here at the 0.1% level, we expect 3 cases by chance when there's no epistasis in any situation, where we have 37 observed cases. So I'm pretty secure that these are showing examples of epistasis. 3 of them are by chance. I can't tell you which 3 are, but I'm happy with that. As we'll see, additive by additive, which had the biggest effect on additive variance, is relatively rare. 15%. Each of these would be 25% if they were all equally present. So the additive by additive is relatively rare. The additive by dominance and dominance by additive is about what you'd expect, while the dominance by dominance deviations occur for 30% of the epistatic cases. So that's enriched for dominance by dominance and reduced for additive by additive. The typical significant effect is 0.15 grams. This is 0.34 standard deviation units. These are the same level of values that we saw for the main effects. So these are many small effects again. All kinds of epistasis are shown. Here's, this is adiposity 8 again, shown as yet a different way. But we can see there's additive by additive. You go down in the blue, you go up in the yellow. You go up here from blue to purple to yellow. Here you're going from blue down to purple, up to yellow. So there's underdominance here, overdominance there, additivity there. So the forms of epistasis are not, when you look at any individual case, it can have multiple patterns. It can be a combination of those different types of epistasis. This is an additive by dominance. You can see that the blue here has got overdominance. Yellow has underdominance. If we go this way, you're going up. This way, we're going down. And this is a dominance by dominance epistasis, where you can see that in this case, the value is high for the double heterozygote and for the 4 double homozygotes, and low for the single heterozygotes. So I think that for the course we said there's going to be a test on these numbers.
Yep,
Yeah.
definitely.
You got to know all of these numbers. This is a more comprehensive view of the epistasis, but it shows the same thing. This is the number of main effect QTLs we found for each weekly weight and the organ weights, tail length and the weight of the heart, kidney, spleen, liver, and fat pad. Remember, there were 8 adiposity loci. So this is the number of single locus QTLs we found. This was the number of interacting pairs of loci we found. Here's that 37 for the fat pad. This is the number of epistatic QTL. And you can see that it greatly outnumbers single locus QTL. There's a lot more genes affecting the trait than there appeared to be. And this is how many of them are new loci that did not have a main effect. So you can see that there's a lot of epistatic interaction going on. And here are some of the interactions, because some locus pairs have more than one kind of interaction. And again, you see here the additive by additive is about 15 to 20%. I combine the additive by dominance and dominance by additive because it's just a matter of the order of loci. And that's about 50% like you expect. And the dominance by dominance is overrepresented to make up for the underrepresentation here. The effect sizes of the values for epistasis and main effects are shown here. This is the average additive genotypic value in terms of standard deviation units. The average dominance genotypic value in terms of standard deviations. And here's again then the additive by additive, additive by dominance, and dominance by dominance. And you can see they're in the same order. They're all small effects. So do gene effects vary depending on the genetic background? Is there epistasis? Looked at in the way we're looking at it, yes, epistasis is common. There's even more epistatic loci than there are pure main effect loci. Epistasis is the rule, not the exception. So this epistasis is going to have evolutionary consequences. And I now count as a graybeard anyway. So do you guys know about any of these? Sewell-Wright's shifting balance theory. Theodosius, you guys know Theodosius Dobzhansky, right? The father of Brazilian genetics. His studies of natural variation in fruit flies were an attempt to show the value of Sewell-Wright's shifting balance theory of evolution. Conrad Waddington's theory of canalizing selection. Schmalhausen's theory of stabilizing selection. Lerner's models of genetic and developmental homeostasis. And also Ernst Mayer's ideas of unity of the genotype and genetic revolutions. And since I'm from WashU, I have to include Alan Templeton's theory of speciation by genetic transilience, which Alan says is totally different from Mayer's genetic revolutions. And so it must be true. This model involves a few loci. The genetic revolution happens all over your genome. So all of these theories, these are the only ones not on this list is Fisher. All of these people thought that epistasis would play a critical role in evolutionary processes. So Mayer's unity of the genotype is that you can't decide the effect of one locus without considering all the other loci. They're unified through epistatic interaction. The genotype isn't a separate bunch of particles dumped together.
but rather a bunch of particles that are all tightly intertwined in their relationships. Genetic revolutions occur when the effects of alleles change. And when, in Meyer's theory, this would be a founder effect. At a founder effect, you take a small population out of a large population, and that small population is going to have different allele frequencies by chance than the major population it arose from. Therefore, the effects of all the genes, all the alleles, would all change after a bottleneck. So these, as Sewell Wright's theory of shifting balance, is that epistatic interactions will lead to differences in fitness of different subpopulations, because each subpopulation has its own background evolved by genetic drift. And then when you get a particularly good combination in one population, that population grows a lot and then sends migrants out to the rest of the population. So only Fisher isn't on here. Who else? George Gaylord Simpson, paleontologist. He used Wright's shifting balance theory in his thinking. So all this was going on in the 30s, 40s, and 50s, except for Templeton, who I won't put him in the 50s. This is the 70s, probably.
Yeah.
But the basic evolutionary thought was that epistasis was critically important. As molecular studies started, and people were able to look at isozymes, different protein variants that separate out on an electrophoretic gel, then they decided that basically almost all variations seem to be neutral. Whereas here, all this epistatic variation is supposed to be important, and important in adaptation. So the whole idea of adaptation in evolution kind of gets lost by the neutral model that came along. And the idea being that most variation in standing in populations has no meaning for the phenotype and has no meaning for fitness. Now, as a morphologist, I see these morphological variations. I can understand that they can lead to functional differences that they could be selected on. I see these species differentiated on these features in relation to their function, their use, and their ecology. So I don't believe the neutral theory is really explaining much of the adaptation, of course, and that adaptation is a real phenomenon. So, I got a vote of confidence from Ernst Mayr. I talked to him about this, and he told me I wrote a paper that was great because I didn't cite his 1963 book. I cited the later paperback that was a shrinking of the massive 1963 book, and thereby got it right according to him. So we're going to look at two evolutionary consequences of epistasis for now. We're going to look at the evolution of genetic variants as populations pass through a bottleneck, and Charles talked about this, and the evolution of genetic architecture, the genetic background under random genetic drift. These studies use the same population of mice that I've been talking about. But in the F3 generation, I'll first talk about the theoretical stuff. You saw graphs like this from Charles. The top left here is what happens to additive variants over the generations with a population bottleneck of size 2, 8, 16, 32, 64. Eventually, given enough generations, all of these would go down to 0 like the 2 did here. And you can see it's just a straight decline. If instead of an additive situation, we look at a situation where there is only additive by additive variance, and again, we start with allele frequencies at 50% like my population has, you can see that instead there's an increase in additive variance to the same level. The 64 would make the same level if we extend it out. What's not shown here is that after the peak, this comes down to 0. So I'm showing these up to the peak. So for a certain number of generations, more generations for a bigger population, you get an enhancement of additive variance while you're going through a bottleneck. You can see that's a substantial one. Additive by dominance over here shows that there's a slight drop and then an increase. And again, these would come down off the board. And dominance by dominance epistasis as well produces additive variance. And you can see this is not a nice exponential line like that, it's bumpy. But this was drawn on a plotter before. There was a time before you could do everything in the computer, so this was a plotter drawing, and that's why in part it's wiggly. So we decided then to look at our epistasis in our F2 population with the real values that we found for epistatically interacting loci and see what happens at those loci. So instead of having perfectly pure epistasis, like the example I just showed, has a mix of epistasis and non-epistasis, dominance and additivity all together. And this shows examples from 4 different locus pairs for the fat pad. And on each one of the graphs, we have the additive variance on the y-axis and generations on the x-axis. The light-colored symbols in line refer to the non-epistatic genotypic values. So this is what would happen when there was no epistasis at all. I eliminated the epistasis. Look at what happens. And you can see typically that you very quickly go up a tiny bit at most and then go down to 0. So dominance has very little role in starting at a 50-50 allele frequency. It has very little role in determining the increase in additive variance, where under epistasis you can see it always is going up and then down. So epistasis increases additive genetic variance as populations go through a bottleneck. These are all done at an effective population size of 4 because I had calculated it out that I expected my experiment was going to use the effective population size of 4. And I wanted the expectations for the experiment to actually come out of these simulations or see how well it did. So the experiment is designed to find this peak, to get the number of generations it takes, which was 7, basically get this peak, which is usually about the 7th generation when you start at 50-50 allele frequency. And here's a whole bunch more numbers. And here what we have are predicted ratios of the F7 generation, that's the 5th generation since we started the bottleneck, divided by the F2 original population. Here's the additive variance using all the genotypic values. Is given the ratio of the F7 to the F2. Now, if things were purely additive, it would always go down. So these ratios would all be less than 1. As we see when we use the non-epistatic value, indeed that's what happens. All these ratios are less than 1. So you're always losing variance when you get to that 7th generation from dominance and additivity alone. Here we have only the epistasis value, genotypic values contributing. And here you can see that for all of these traits, the ratios are way over 1. So the epistasis is producing new additive variance as you go through the bottleneck. And this column is all these two together, basically, in average. So you can see that the effect is not totally washed out when epistasis is added in with the additive and dominance. So we have a big inflation, 3, 4, 8 times of the additive genetic variance as populations pass through a bottleneck. This just shows the ratios above and below 1 when we're looking at populations through the bottleneck. This is the ratios with the genotypic values. You can see that the ones above 1, which have enhancement of variance, are much more common than those below 1, except for 210 weight, so they're about the same. And then if we only look at the non-epistatic values, remove epistasis, we get like the opposite result. So basically, it's showing that epistasis leads, natural epistasis that we find in our population leads to enhanced additive variance through the bottleneck. This is the same as the other slides. The other slide was, except instead of starting at 50-50, we started our population of 8 individuals at gene frequency of 0.125 and 0.875. So now we have a rare allele instead of all alleles having a 50% allele frequency in the original population. And again, if we have here, the peak is in the F5 generation, not the F7. So it happens faster. The enhancement happens faster, and then the loss also happens faster. And here we can see again the ratios for the non-epistatic values, and they're pretty small. This is for the epistatic values alone. You can see even when the allele frequency starts at 1 out of 8, the lowest allele frequency you could have, you still get this massive enhancement of variance through epistasis, and that carries through to the genotypic values as well. So we not only, so we started this inbreeding in the F3 generation. At the same time, we have the outbreeding line I talked about yesterday. So from the F3, 1,600 F3 animals, we kept a random mating string. We actually kept 2 strings with effective population size greater than 125. Those strains got mixed after this F7 experiment. Well, we used this effective size of 4 randomly and got to the F7 generation, and then reared up a bunch of families at the F7 generation to see what happened to the additive variance compared to the F3 and compared to the control strings. Now, since we did this randomly choosing 4 individuals without replacement, we had variation in the inbreeding coefficient, because when you're randomly selecting 4 animals, they're really coming from 2 families. So some lines will have more full sib matings, and other lines will have less full sib matings. So they'll inbreed at different rates. There'll be variation in the inbreeding coefficient. And that's shown here. And the average inbreeding coefficient predicted for the 7th generation was 0.4, and we got a 0.39. So I was happy enough with that. And you can see that it's not quite normally, it's maybe log normally distributed, which I think is what you'd expect for the inbreeding coefficient. So this is a plot of the inbreeding coefficient against the additive genetic variance for each of a series of 39 different populations. So 39 different populations, each undergoing the same kind of bottleneck. And then we measure the additive genetic variance in each of those populations. This is done quantitative genetically using family membership, not using QTLs. And you can see this is a graph showing the additive genetic variance. You can see that many populations don't have any. Some of them even have negative. It's not really allowed, but estimates happen that way when it's really 0. So, and you can see a lot of them don't have any genetic variance at all. The dashed line is the expected level. However, you can also see that there are a lot of populations above and way above the expected. If you treat all these populations together and get an average additive genetic variance to look at, here we have the data on the additive genetic variance in the control lines, and then in the experimental lines. These are the inbred lines of the F7. The number of animals in the total samples, the average number of animals per sieve ship. You can see that it's bigger, 9 for the control lines, only 7.5 for the experimental lines, because there is inbreeding depression for productivity. And then here's the additive variance calculated for the control line and for the experimental line. And you can see the experimental line has a higher variance. These are the standard errors of the two variances. If we compare this experimental variance to the variance you would expect given the control line additive variance and the amount of inbreeding that we produce, your expectation is that the variance would be 5.18. This is for adult body weight. And our observed is 9.11. So we got nearly a doubling of additive variance compared to what was expected under an additive model. And this is statistically significant, not massively so. And we also did this test with bootstrapping, with jackknife, and it's significant no matter what we do. But it's not highly significant. The only way this would have been highly significant, even though it's almost a doubling, it's a borderline significance, was if I had more than 39 lines. Well, I started with 55, and 16 of them died out before they got to the F7 generation, due to inbreeding depression, due to lack of fertility, and also mostly because the mothers wouldn't take care of their kids. That was the primary reason. So this is an experimental validation of the models that Dr. Grimlight was talking about. Here we actually see additive variance rising as you go through a bottleneck. You can also look at the relationship of the deviation from additive expectations on the y-axis and the deviation of F from 0.39, which is the average. And here you can see a very strong relationship that's not expected under additivity, but would be expected under epistasis. It would have been better if I had fixed the breeding of all 39 of the lines so they all had an inbreeding coefficient of 0.4. I would have then gotten an even bigger distinction between the expected and the observed. So there's a lesson I learned. Wouldn't follow the model directly, but it would give you more power to show the enhancement of additive variance. If we looked at all of our traits together, you see the same phenomenon, that the expected additive variance over all traits is enhanced. And the probability of that at 0.013, this is for weekly body weights. The first one was for week 10 body weight by itself. This is all the body weights. And then likewise for the fat pad, here we see this big increase, doubling and more of additive genetic variance going through the bottleneck. Turns out the different traits have different amounts of epistasis and have different amounts of enhancement as they go through the bottleneck. So the average additive variance increases or at least remains stable during inbreeding rather than declining as it should under an additive model. The enhanced variance effect of inbreeding is greatest at intermediate levels of inbreeding, about 0.4 for the average inbred coefficient. The other thing is bottleneck experiments must include a very large number of lines. So back when these models were first produced, recent effort that Charles talked about had a result where she had a line and it increased in additive variance. And she was, Ed Bryant was the other author. We were all very excited to see that because it actually gave credence to Charles and Mike Waite's crazy ideas. But it turned out she was just very lucky. As you could see, probably more than half of my populations decreased in additive variance. It's only the average that went up. So if I randomly picked one of those 39, I most likely would have found no enhancement. Unless I was lucky and picked one of the ones that was at the top of the charts by chance. So there was a paper written in 1988 by Mike Lynch which stressed that point, which I then tried to follow. So we're also going to look at another study that's related to the local average effects that Dr. Goodnight talked about. We read his paper and we were just so beautifully written, so clear and understandable. And you could see the experiment is right there. He lays out the experimental design beautifully. So as we were inbreeding these strains, we went on from the F7 generation to a later generation of F16. And by this generation, the inbreeding coefficient is 90%. This was work done with Professor Reinaldo Alves de Brito, who you may have heard of. So this is going to be about the evolution of the genetic background under genetic drift. So do the effects of genes change as genes differentiate under genetic drift? So not just does the frequency of the gene change, but does its effect change? Because frequencies change at other loci. So no, under an additive model, the means only diverge, the backgrounds do not diverge, because there is no real background effect. Yes, under a model including dominance and epistasis, both the means and gene effects diverge. So, and do alleles change their order of effects in different genes, so that a gene making an individual larger in gene A makes them smaller in gene B. So not only is there differences in the amount of effect, but does the effect actually switch from positive to negative, which is the most powerful form that epistasis would take. So here's the breeding design all the way down from the F4 to the F16. We were down to 26 lines instead of 39. Average inbreeding coefficient of about 0.9. So we had a series here of 26 recombinant inbred lines. These lines have since gone to, some of them are in the 50s, 60s generation, and we're down to 10. Then I made a whole bunch of new ones, so there's now another 35 RI lines. And we took these RI lines and we crossed them with 4 different inbred mouse strains: AJ, AKR, C57 Black 6, and DBA2. These strains are chosen to represent a wide diversity of body sizes. So instead of randomly picking strains, I purposefully picked strains that were of different body size. AKR is big, A is small, Black 6 and DBA are intermediate. And I mated them into each of the 27 lines, producing 16 F1 hybrids for each combination. And you can see here the total number of animals, 1,728. This is the model suggested by Charles, where the phenotypic value of the trait is equal to the mean, plus the effective strain. So this is the RQI strain. These strains are experimentally set to have a large difference by using strains with inbreeding coefficient over 0.9. We've left them drift apart quite a ways. If we'd done this study in the F7, they wouldn't have drifted quite as far apart, and their backgrounds probably wouldn't be very distinct. The second term here is the outcross strain, the AKR, Black 6, and DBA2. And this was experimentally set to a large effect by using strains with diverse body sizes. So these two terms can be statistically significant and not be really meaningful to us because that's the way the experiment was set up. The term of interest here is the interaction between strain, between RI strain and outcross strain. Because here, it's the strain interaction. This is the factor of interest. Does the effect of having one of the 4 outcross parents change depending on which RI strain it's made it into. This would show variation in the genetic background across the strains. So that was our object of interest. And whenever we talk about epistasis or any interaction, there are really 2 kinds of interaction. One kind is interaction due to variance differences, or differences in the amount of spread. Here we can see the 3 demes are pretty close together. And here we see the 3 demes are very far apart. So but the order of the demes is the same. It's 1, 2, 3, and 1, 2, 3 in both instances. It's just that there's a big variance here and a little variance there. This has evolutionary consequences, but they're not very severe. Other kinds of interactions are due to order differences, and this is the crossing, like I showed you. Looks just like the additive epistasis. It's a crossing interaction. Here, the actual effects change order, so when you're mated into the first deme, you're high, and the first deme is high. When you're mated with the other, you're low. So the order of effect of the demes changes. In this situation, when you look at 2 populations, the large allele may make you larger when, or the AJ allele may make you smaller when mated into RI strain 1, but make you larger when mated into RI strain 3. It'll change its effect from being a positive to a negative going across the strains. So this interaction by spreading will produce different rates of evolution, but the process will be the same. You'll have a high rate with this situation and a low rate over here because of the variance differences. Here, though, the evolutionary consequences of crossing is that different alleles would be favored by the same selection in different demes, leading to genotypic differentiation in the face of phenotypic uniformity. So here, if we select on these 2 situations for a bigger size, we would favor different alleles. Here you would favor the capital A allele, here you would favor the small a allele. So the allele that makes you bigger and is favored by selection will change from deme to deme. These results are presented in 2 ways. The correlation of local breeding values, which is the variance between strains divided by the variance between strains plus the interaction variance between strain and outcross. That's going to vary from 0 to 1. A 0 indicates a strong interaction and a 1, no interaction. We can also have the correlation of the recombinant inbred strain means across outcross strains, and that's going to vary from -1 to +1, with lower values indicating more extreme order differences amongst the strains mated to the different outcross ones. Here's the table, and if you look in the paper, which is like 2005, you won't see these beautiful probabilities. So the probability of no effect for strain is 2.9 times 10 to the minus 147th. For outcross, it's only 10 to the minus 108th. Of course, we set these to be high and strong. But here our interaction term, the one we're interested in, multivariately comes out with a 10 to the minus 112. So not only is this present, but there's a lot of it present. They said that these were all too extreme, and we just had to say they were less than 0.1 or something, or less than 0.01.
0.01.
You know, yeah, they're less than 0.01. They're less than 0.100. So I like these probabilities, but the journal felt that they were bragging. So I don't question any of the statistical significance here. So there's significant interaction variance, meaning there's significant variation in local breeding values. This is the first form of correlation where the low number is more interaction and a 1 is less interaction. And you can see That's varying here from 0.2 to 0.7. So there's a fair amount of interaction measured that way. And this is the correlation which would go from -1 to +1. And you can see that it's likewise is running from 0 to 0.265. So for this correlation to have gotten negative, there'd have to be mostly crossing variation and no spreading variation. That's the variance portions for males. This is for the body size traits, the weight, the tail length, heart, spleen, liver, fat, head, and kidney. And here it goes, only -62 here, so barely significant. But again, you can see the range of values going from 0.3 to 0.6. So a fair amount of epistatic interactions, or a fair amount of differentiation in local breeding values, and the correlation running from near 0 here to 0.28. The lowest value here you can see is for the fat pad, which is why some of our work is concentrated on the fat pad. Going back to the original epistatic studies, we found more for fat pad than for others, more in surrounder epistasis. So here's, so this is a chart showing the results for male week 1 body weight, which is a place where there was a lot of variation in breeding value, local breeding value. And here what's plotted is each one of the outcross strains and their average phenotype with each one of the RI lines. So there's 26 lines in here. You can see some matings never worked out. This was one of these figures. That RI line only mated with AJ, and see there's 7 lines that failed with these 2. But they fail a lot just for fun. There's nothing really much to be mated in. And you can see that there's a lot of crossing. What you're supposed to look for in here is crossing. And there's a whole lot of crossing going on. But this includes both spread. You can see the spread is big mated with AJ compared to what it is mated with LAC6. So there's also this spreading difference going on. And we were able to remove the spreading difference to standardize it so the spreading difference is the same for each outcross. And now all of the things you have here are order interaction. And you can see there's substantial crossing of these lines. Even for a trait that showed a lot less interaction, we still got lots of crossed lines. When you remove the spreading interaction and just go with order interaction, here you can see there's a lot more parallelism. Because this is one that had a lower amount of variation in local breeding values for female 8-week weight. So those are the 2 extremes I'm showing. So you can see here, you can kind of see a lot of this parallel. So the conclusions here are the genetic backgrounds of the independently involving genes have diverged substantially, often differing to the same extent as gene means. So drift is not only affecting the frequency of alleles, it's affecting the effects of alleles. It's changing. That's the genetic revolution that Meyer is talking about. Genetic revolution due to the unity of the genotype through epistatic interaction. The effect of introduced genes varies from one gene to the next, so that common phenotypic selection on the genes will produce genotypic divergence underlying phenotypic uniformity. So if we have these different genes, they're not migrating between them, but they all came from a common source. Over drift, over time with drift, the effects of these genes will change. So even if both gene has the exact same selection pressure exerted on it, say for increased size because the temperature everywhere is getting colder, something like that, different alleles, opposite alleles, would be favored in different genes. So these 2 genes with opposite allele effects, you'll fix one alternate in one population, the opposite allele in the next population, even though the selection was identical. eventually you build up enough unity of genotype in this new configuration as populations get bigger, that when you then go and hybridize them again, even though they've been under the same selection and pretty much look the same, there is hybrid dysgenesis and the formation of incipient species. Without any ecological cause here at all, the ecology of the two is the same, the selection on the two is the same. What's changed is the effects of the genes, the effects of the alleles. What made you bigger in one population makes you smaller in another. So this conditional situation is critically important. And we hope that the individual genomic regions responsible for these interactions can be identified. And I have to get DNA out of these guys again. Then Reinaldo is going to have them snip typed somewhere. And then we'll be able to tell you which regions of the genome are most likely to interact. So I'll finish off with a couple of quotes. This is another one from history, a great biography of Julius Caesar by Meyer, translated by McClintock here. It's written in German originally. And he's talking about the role of the individual in history. And we must then consider not just the personalities, their general and particular interests and opinions, but the positions they occupy within the configuration. So Caesar can't be considered alone, he has to be considered within the context of what's going on in Rome at the time, which is of course determined by their action and interaction, but also determines them. It's a two-way street. One must therefore consider not just the participants, but the total situation, which is greater than the sum of its parts. True, such an approach makes it difficult to form one-dimensional judgments with scholarly detachment, but it brings us closer to the reality. So, I probably shouldn't say this, but molecular genetics. Right there. And then finally, from Lewontin's book, The Genetic Basis of Evolutionary Change. This is the very end of the book, and you may know he's a diagonal socialist. So his example comes from there. The fitness of a single locus ripped from its interactive context is about as relevant to real problems of evolutionary genetics as the study of the psychology of individuals isolated from their social context is to an understanding of man's socio-political evolution. In both cases, the context and interaction are not simply second-order effects to be superimposed on a primary network, a primary monadic analysis. Context and interaction are the essence. This was, not a too uncommon view in the 1970s, because we were all brought up on Meyer, Dojansky, and Wright, and Fisher. But we were always told Fisher was wrong. I had my PhD at the University of Wisconsin-Madison, where Sewell Wright was still there. I got to talk to him about my thesis. I don't know if I made any sense or not, but I had many opportunities to talk with him. This then shows during the time that this was going on in the lab, this is from 2000, all right? So this is 12 years ago. And Andrea looks just the same. Although used to have black hair. This is Susan Pletcher, who's been the lab technician for the mice for 20 years. This guy, you'll see in a little bit. This is Jason Wolf. And he was there at the same time.
He's just the same too.
So I managed to get all three of you in one shot. This was, those favorite other postmen. They would fight over the good computer.
This is on YouTube, remember.
Well, they would fight over the good computer, and one would get up and leave the computer and go do something, the other one would jump on. I finally solved that problem in the lab. Now the best computer's in my office. I used to put it out where people could use it because I never did anything but word process. Everyone else actually didn't do the work. This is Elizabeth Norgard. These are mouse small and a large mouse cupcakes. See ears and tail, and large is white and small is black. This was Elizabeth's 16th birthday. Last year she got her PhD in my lab, and is now in Maine, in the U.S., teaching at Colby College. So she got frustrated and came back. She knew she could do something with us and be done. So, well, thank you for all that. I know that I went on a long time, didn't I? So we can take questions, or you can come up and we can talk as we break. Trying to see what time it is now. It is 10:30.
10:30.
10:30?
Yeah, about that.
Well, that's me going on a long time. So we'll take a break now and then come back for more simple mathematical models.